All right, guys. So thank you again, Isaac, for inviting me. Thank you all for having me. I'm really happy to be here and excited to share the work that me and my team have been working on. So we're specifically interested in extracting knowledge about COVID-19 treatments from scientific literature. Brandon already gave a really great overview about both CoronaWhy and this CORD-19 competition. So I'll just go into it a little bit more. This is a Kaggle competition that was organized by AI2, Chen Zuckerberg, Microsoft Research, the White House, and they kind of put together this big experiment just to see, okay, what would happen if we release a bunch of open access papers to a bunch of Kagglers, so that's data scientists, machine learning engineers, and see if they can extract knowledge from these papers related to COVID-19 in a bunch of different facets. So it was very much unlike the typical Kaggle competition of, all right, here's the input, here's the data, output, here are the benchmarks you're trying to beat. It's very much kind of open fields, and it's kind of very exploratory. So they have 10 core tasks, and one of the tasks, and the one that I was focusing on, was what do we know about vaccines and therapeutics specifically? And this is because I come from a computational pharmacology background, so this is very in line with some of my previous thinking and my previous experience. So even within each of these tasks, there are some subtasks, but the subtasks are also still pretty general. So things like, what's the effectiveness of different drugs being developed and tried to treat COVID-19 patients? So I see this as a great kind of creative opportunity to just try to extract knowledge about drug treatments in a bunch of different facets, and kind of that result is going to be very useful for a variety of different people who would be interested in quickly summarizing the literature. So the context for this whole challenge is that there's so much new scientific literature being put out about COVID-19, both in terms of preprints, so things that are not yet peer-reviewed, and even published papers. So just being able to kind of distill like the latent key knowledge that's actionable from that whole kind of like inundation of literature, because obviously COVID-19 is on everyone's mind. That's kind of the big value, and that's what we're trying to do with CoronaWhy, and that's what they're trying to do with this Kaggle competition. So just to give a little bit more detail about what is in the CORD-19 dataset. So it's a bunch of these papers. Most of them are full text, but some are just abstracts, and they come from PubMed Central or PMC. PubMed Central is kind of this repository of scientific literature that exists. So papers are indexed, and you can find abstracts and often full papers for scientific literature across a variety of journals. Then there's also papers that come from Elsevier, so that's another like publishing house. And then we have some preprints, so things that are not yet peer-reviewed, that are coming from bioRxiv. So that's going to be kind of more like experimental literature about people doing work on kind of cellular screens or maybe computational work. You're going to see papers from medRxiv, or preprints from medRxiv, so that's more like clinical kind of literature, and from just kind of regular archives, so maybe more algorithmic kind of stuff. The query that they use to get this set of papers, which is kind of in like 40,000-ish papers, is a really, really simple query, just looking for terms like COVID-19 or like SARS-CoV-2 or just related coronaviruses. So really MERS, SARS-1, and SARS-2 are the coronaviruses that are represented in this set of papers. So a pretty simple query, but they just kind of, AI2 is hosting it and putting it all together for us to analyze. So my team specifically is the one focusing, like I said, on vaccines and therapeutics. And I've had really, really a great fortune to work with such an awesome collection of people. So people coming from very diverse backgrounds, like data scientists, NLP engineers. Ben, who's with us, is a professor in physics. We have people who are pharmacologists and virologists and data visualization experts. So I've been really, really privileged to work with such a great group of people. And I'm going to show you what we've been working on and what we're thinking about for the future as well. So this competition is happening in 2 key rounds. So the first round just finished a couple weeks ago, and I'm going to talk about what we did for that round. And the second round ends in a little over a month from now. So there's not much continuity between the rounds. It was more just like the Kaggle judges are evaluating the submissions at 2 different time points, round 1, round 2. And the tasks are staying pretty much the same between the 2 rounds. All right, so round 1. We were interested in this round in getting just kind of a landscape of all the drugs that are being either currently used for repurposing or considered for COVID-19 treatment. And we wanted to kind of summarize those drugs that are being mentioned in the literature and present them in kind of a dashboard that a user could quickly query through. So what's the issue here? The issue here is that there's no drug that exists that has a label indication for specifically COVID-19. You know, nobody's developed a drug saying, okay, yeah, use this to treat COVID-19. And to create a new drug takes a long time and a ton of money. And we don't have that kind of time right now because we're finding ourselves in the middle of this pandemic. So what people are doing instead is trying to use existing drugs in an off-label way to treat this, this disease. And so you see, for example, chloroquine, hydroxychloroquine are like a couple of the main repurposing candidates. And those were also antiviral and anti-malarial drugs. But it's very common now that people are trying to look for other drugs that already exist that could be useful for COVID-19. And people are looking for repurposing candidates at all stages of the clinical, of the research pipeline. So doctors are thinking, okay, maybe this drug could work, maybe that drug could work. But also even experimentalists are saying, okay, I have this kind of model cell or this model organism that would be a good representation of how the virus is transmitted and enters into cells. Let me see if this virus blocks this one little key function. Or even further back in the pipeline, you might have computational researchers who are doing kind of simulation-based studies to see, okay, this little small molecule drug should bind to this protein receptor, and this protein is really important for viral entry, let's say. So a lot of people are thinking about drug repurposing kind of scattered throughout the literature. This kind of tool that we were creating could be useful for researchers who are interested in doing kind of early studies about potential repurposing candidates and to kind of connect efforts between one lab and another lab because they both have different kind of orthogonal studies on the same drug. And it could also be useful for someone who's maintaining like a clinical database curator. So they would be interested in seeing like, okay, what's the latest research about chloroquine? Let's update a couple of facts about chloroquine in our database. Now, we stress that the point is not to give direct kind of clinical recommendations. The point is just to summarize and quickly distill the literature, and then someone else who has kind of the clinical expertise, they can go in, they can do the due diligence of looking at the studies, all the qualifications and the context for the studies, and kind of use that to inform clinical practice. But we're not trying to make any recommendations directly. So the methodology for this is relatively straightforward. First, we wanted to query sentences that mention both a drug and a trigger term that either indicates that the drug is being used for treatment or used for or considered as a repurposing candidate. And we just looked for sentences where both a drug and a trigger treatment term co-occur in a sentence. And then we considered a couple of filtrations from that because that's a very simple kind of way to query sentences at first. First, we considered removing sentences that contain a negation, and by negation, I mean a word like not. So for example, if a paper or a sentence said, we do not recommend hydroxychloroquine for treatment, that we want to try to remove those kinds of sentiments. We also considered using a BERT-based sentence classification model. So BERT is kind of the latest and greatest supervised, this kind of massive deep learning model in natural language processing that's taken the world by storm. So we wanted to see if we could use this as a way to classify sentences as true drug treatment sentences. And then we wanted to be able to give the user the option to filter out to those papers that do actually concern COVID-19, or maybe you would be interested in other coronaviruses as well and the diseases related there. So we leave that as an option for the user. And then create a visualization. So the drug lexicon that we use, this is the vocabulary of drugs, and it comes from RX norm, which is a very standard and a pretty comprehensive set of drugs. There is a couple of kind of noisy things about this lexicon. So for example, there are periodic table chemicals, there are some random animal names and fruits and vegetables. So we had to kind of clean out some of the garbage, and that's a standard bioinformatic pre-processing, part of the job. We also incorporated a few lexicons from kind of modern therapeutics that are being considered, and some more research chemicals that wouldn't be captured in this RX norm lexicon. So there's WHO data, and Xclara has a database as well. So we want to make sure that we're trying to be as thorough as possible. We looked for a list of words that indicate some sort of treatment. So literally words like treat, treatment, other words about like how the drug is administered, and also words like repurpose in the case of more like experimental or computational studies. Now, we were interested in human annotators data for a couple of reasons, for both classifying sentences at the sentence level, if it's a true drug treatment sentence, and also trying to see if we could get a sense for negation and remove those negation sentences. Now, we were also dealing with a time pressure, and so we didn't have time to get a lot of annotations and do kind of rigorous annotation testing. So what we had here is a set of 508 annotations from a single curator that split sentences into either yes, the drug and the treatment sentence, drug and the treatment word indicate that the drug is actually being used for treatment, or no. An example of no would be something like, I gave my dog some treats and the treats were next to the Advil on the counter. And that's not talking about Advil as being used as a treatment, but that's kind of that's a false positive. We want to remove those kinds of sentences. And then we asked the annotators, okay, if the drug is being used for treatment, is the fact that it's being used for treatment negated? This was a bit of a tricky request because the intention was more like purely syntactically negated. So this is not a treatment. But what people were kind of turning up was also kind of more semantically, this is not like an effective treatment, because maybe this drug treatment leads to adverse drug reactions, for example, and people were kind of labeling that as, yes, it's negated, even though that's not kind of canonically like a syntactic negation. So take the results that I'm about to show with a grain of salt, that the annotations, we could do another round of annotations that would be higher quality. So we considered a few different ways to do negation detection. For one, we looked at this list of just negation trigger terms. So things like no, ruled out, no evidence, etc., that are listed here on the right. We considered using dependency parsing. So that's kind of looking at like the actual linguistic sentence structure and seeing if the negation occurs along kind of a dependency path between the drug and the treatment trigger word. And then we use this other neg spacing model. So that's just kind of it's negx, which is a rule-based system, or kind of like a proximal, certain negation words will occur near the treatment trigger word, but a kind of a slightly different set of negation triggers than the first one. So we ended up going with the set of negation trigger terms that we came from this like latest paper here. We just got kind of very slightly better F1 scores for that. And again, for this stuff, we keep it as an optional filter so that the user can choose whether or not they want to incorporate this feature or not. Again, given the limitations of the annotations. We also did the BERT-based sentence classification. So that was trying to classify here, yes, the drug is being used as a treatment, or no. And so we did this using a Roberta sentence classification model. So this is just a BERT model, but they've done a little bit of hyperparameter tuning. They've optimized the BERT model even a bit more. And so with these annotations, and these annotations are fairly reliable between whether the drug is being used for treatment or not. And so we got decent recall on this set when we looked at training and val. And we did sort of just a qualitative analysis for testing. So again, that's a limitation of the annotations. One more thing we were interested in was identifying the study type specifically. So there are a few categories we're interested in silico studies, so like a computational kind of a simulation-based study, in vitro studies, which means like little like cell culture was used, in vivo meaning an animal model, or clinical studies broadly. So actually somebody is using this drug as a treatment within a hospital setting. For this first round, just kind of as a first pass, we created vocabularies for each of these 4 study types. And we classified a study based on the study type, based on which vocabulary was kind of most present in the study. So this is, again, just a kind of a heuristic first pass approximation to identifying study type. But qualitatively, this was quite good for our purposes. So let me go to kind of a quick demo of what we created. So this is the dashboard that we created using Power BI. So this is a Microsoft kind of data visualization product. And what it allows you to do is you can search for a specific drug. So let's say we're interested in remdesivir. We see remdesivir and we see the number of studies where remdesivir is mentioned as a treatment. And we can select based on the study type. So maybe we're interested in only clinical studies. And then we can look for specific sentences where remdesivir is being used as a treatment. I can click the sentence and then I see kind of the context. So this is like the paragraph where that sentence was mentioned, and the abstract and the title of the paper that it comes from, with a little bit of data about the year, the journal, etc. Now this little match quality box here lets you choose. Loose means just getting those sentences with a drug and treatment mentioned. Medium means it does that negation detection based on if there's the detection of one of those negation trigger words. And tight indicates both negation detection and another layer of BERT being used to classify sentences. So it'll throw out sentences that BERT said, nope, this isn't a real drug treats sentence. So this allows the user flexibility to explore the literature quickly and see how is the drug being mentioned and how is it being used or considered for treatment. So that was awesome. That was progress that we were able to do in a really relatively short period of time, and a great team effort. So that was really awesome. Ben did a ton of hard work on that, and Ben's here. So hey, hello, Ben, and thank you for all the hard work on that one. A couple things that we would do for next steps is, again, work on those annotation quality issues. We would we would actually get multiple annotators to annotate things and then calculate inter-annotator consistency. We would further break down clinical studies into further study designs, and then automatically classify study designs rather than just looking at vocabularies. And this is something that we're working on already. We've got a large volume of annotations from a bunch of medical students, and we're excited to start automatically classifying study design. So that was round 1. That was kind of the big project for round 1. And now for round 2, we're trying to pursue a few different projects in parallel. And so just in the next couple minutes, I'll just very quickly kind of give sort of the high level thinking for a couple of these projects. So the first one is SARS-CoV-2 mechanistic modeling. And so what we're trying to do here is extract essentially a knowledge graph of all the mechanistic relationships that are being mentioned in the literature that are relevant to viral entry and viral transmission. So we're interested in relationships between proteins, genes, drugs, and then cellular functions. So a drug targets a protein. This protein leads to downstream effects where it kind of signals things to other proteins. Maybe those have effects regulating genes, and a set of proteins all kind of has one cellular function. So you can think of this all as being built up into a big network representation. What we can do is we can take these, sentences that are mentioned in the literature, something like this here on the right. And an example of what a pipeline could look like is you have a sentence here, you identify kind of the key entities in the sentence. Maybe you have to do some anaphor resolution if there are pronouns, but yeah, so then you identify the key entities. You can identify the trigger terms, so like the certain specific kinds of relationships. So specifically, PIV3 is being targeted, and then there is degradation here. So those are kind of the key relationships that are being picked up on. And then you can represent that in these little kind of predicates or kind of triples of entity, then you have the relationship word, and then the other entity. And that's kind of one link within the knowledge graph. And you can use those all together and you can build up kind of a more complete knowledge graph of how that all plays together. And once you have such a knowledge graph, what you can do is you can start to ask questions using kind of a probabilistic graphical model, such as, given that we observe downregulation of ACE2 and IL-6 amplification, what would happen if we were to, with a drug, inhibit this IL-6 receptor? So this is a project being led by Jeremy Zucker in our team, and this kind of lends itself to this sort of causal modeling framework a la Judea-Pearl. So that's a really cool way to extract knowledge from the literature. And we're learning all these new mechanistic interactions every day, and so being able to, in almost like scientific real time, use that to update this sort of network representation, that's going to be very huge, and it's very exciting. Another project we're pursuing is adverse drug reactions. So you use a drug, and often the drug might have undesirable effects. And so we want to be able to detect which adverse drug reactions are being mentioned for these drugs being used in the literature. And in a similar way to in round one, present these in kind of a dashboard so that you could see, oh, these are the things that are being, the kind of general trends we're observing for different treatments. This would be important for, again, updating that kind of a database, or for a clinician or a pharmacologist, or a pharmacist, excuse me, who is prescribing drugs for COVID-19, and just checking that no unacceptable adverse drug reactions might occur based on what's been reported already in the literature. So we're looking at this lexicon. This is the common terminology criteria for adverse events. That's a lexicon of adverse drug reactions. And kind of as a first pass, we can look for co-occurrences of drugs with this adverse drug reaction. But we can also use a BERT-based model to classify sentences more automatically, and that's a more sophisticated way to do it. So this is the BERT model that we're considering, and it's called a span-based joint entity and relation extraction with transformer pre-training model. So that's all to say that we're interested in extracting simultaneously which entities are referring to either the drug or the adverse drug reaction. And at the same time, we're trying to classify, yes or no, this drug is somehow leading to or related to this adverse drug reaction. So this thing is span-based as opposed to traditional, like one token at a time, which is kind of interesting because for some drugs, you might have like, they might be pairs of tokens together, or even like triples of tokens. And so looking for, rather than just like one little token at a time, maybe you want like 2 or 3, and that becomes the interesting like drug entity in the first place. So this is the kind of model that we're considering for that, but we're very much in early phases of exploration at this point. And the final little project that I want to talk about very briefly is extracting and kind of bringing to light contradictory claims being mentioned in the literature. So are there contradictory claims about either drug efficacy, about adverse reactions, or other facets of drug-based therapy that we're seeing in clinical literature? So this is interesting because very recently work was done on, first of all, even just going from a paper, specifically an abstract, and extracting like which sentence in the abstract is actually like the golden nugget where they're like, yes, this is the highlight of this result of this paper. This is kind of like our key takeaway and our key interpretation. And so there exists this LSTM-based model for going from an abstract to actually retrieving this claim sentence. So here's an example of one claim sentence for a paper that was extracted. What we can do is first we can go to a paper and we can try to extract those claim sentences, and then we can apply BERT models, or kind of these sorts of these automatic supervised approaches for extracting if this claim and this claim is contradicting each other. And there's this nice data set called the MED-NLI for natural language inference. And they've already annotated like if this is the premise and this is kind of the hypothesis, do these things entail each other? So does this follow from this, or is there a contradiction between these two things? And we can treat one of the claims as the premise and the other claim as the hypothesis. And then we can use the models that were trained using this MET-NLI data set to try to find those contradictions. So that's a really cool kind of meta-scientific direction to go. We're trying to see if these papers are kind of butting up against each other, because one of the interesting kind of the holy grails of meta science is when a new paper is published, where does it fit into the current body of scientific literature? And this sort of project starts to get at the first question of, okay, does this new paper contradict any other paper's key claims in a corpus? So those are that's my time for now, and those are some of the projects we're working on. So I want to thank you guys for inviting me.